Do I have the camera on? I do. So I had a question, and it's a very common question, and it's a concern. People say, well, when I watch your video, I can't see the pointer on the screen. All right? And it's a very common concern. And while I will admit to you that the pointer is small, there is a way to see the pointer on the screen. So I'm going to show you how to do that because it's actually fairly straightforward. There are two things you have to do to see the pointer on the screen. If you don't do both of these things, you won't see it. Okay? So both of these are necessary. Number one, you have to change the resolution to 1080p. Okay? If you don't do that, the next step isn't going to make any difference. So you change the resolution to 1080p, and when you do that, then you click on this full screen thing. And when you do that, you will get the full resolution and the full screen, and that should bring the pointer online for you. Okay? If you try to view it as that little image, you're not going to see the pointer. It's just too small. But if you just try to enlarge it and you don't change the resolution, you won't see it either. You've got to do both of those things. Everybody with me? Do both those things, you'll be in good shape. Okay. So let's get out of that and into our fun topic of the day, which is protein structure. And I know you guys are just dying for protein structure. And we're going to talk about protein structure. And I hope that by the end of the lecture today, you're really beginning to appreciate the really cool things about protein structure. Okay? So last time I introduced the topic of tertiary structure. And tertiary structure is what we call the folded structure of a protein. Now, fibrous proteins do not have tertiary structure. They don't have folds. They just have fibers going on and on and on and on. Okay? That's an important consideration. Folds happen, you recall, because of turns, and turns cause those helices to sort of orient themselves into a folded shape. Okay? So folding is essential for a globular protein to have its proper shape. Whenever I say the word or say the phrase globular protein, you should be thinking of tertiary structure. You should be thinking of folding. Okay? Because only a globular protein will have tertiary structure and will have folding. Okay. Well, a really good example of a protein with tertiary structure is commonly used is myoglobin. That's not myoglobin. That's part of myoglobin. And myoglobin, oh, come on, Ahern. There's myoglobin. I showed you that the other day. All right? Myoglobin has, in this case, mostly alpha helices with some turns. And you can see this other thing in here, which is what those other structures were showing you. And I'll show you that in a minute. But the important thing about this protein is it's folded. And it's folded as a result of those turns. And the folded structure of this protein, even though it looks random, is actually quite precise. If we disrupt the folding by doing some things to it, and I'll describe those things to you in just a little bit, then it will no longer stay in the folded state, and it will no longer continue its function. The function of this protein is grabbing a hold of oxygen and holding on to it pretty tight. It's related to hemoglobin, but it's not hemoglobin. Okay? So it grabs onto oxygen and holds onto it pretty tight. You can see the oxygen, those two little red atoms right there, and they're being held by this odd-looking structure here called a heme, which I'll show you in just a minute. Okay? That's tertiary structure. Now, the heme is the functional part of myoglobin. It's also the functional part of hemoglobin. It's what gives hemoglobin its name, and both myoglobin and hemoglobin contain heme. All right? The basic structure of heme, okay, is something called protoporphyrin. Protoporphyrin is this complex of ring structures that you can see right here. And if we put and iron in the center of that, we get what we call heme. So this is a sort of support structure for the heme, and the heme is what happens after we put everything together, which includes the iron. Okay? The iron is actually the part of the heme that holds onto the oxygen. The iron is the part of the heme that holds onto the oxygen. It's this structure here that was that odd-looking structure back here. That's actually heme right there. You can see there's an iron, well, you actually can't see it, but there's an iron in the middle of that, and it's holding on to an oxygen right there. Heme, yes?
You mentioned before that in other organisms that instead of it being iron, it's say copper or other things. Is it
Yeah, good question. Okay, so good question. Her question is, if it's not iron in there, is it still heme? And the answer is no, it's not. So only if we have iron there do we call it heme. In plants, there's a very similar structure, and in the middle of it, they have magnesium. Okay? And you know that structure as chlorophyll. Chlorophyll and heme are related to each other. Okay? Or at least the functional part of them is related. That is the part that holds on to, in this case, the oxygen. So if we were to have chlorophyll, we would have a protein that has, let's go back here. We would have a protein that has this structure, but instead of having an iron in there, it would have a magnesium in there. Okay? All right. Now, if we look at this heme structure a little more closely, we can see over here what it looks like. Okay? Here is the heme, and we can see that it's a sort of a flat structure. It's got these flat ring-like things. In the middle, there's that iron, or that iron atom. All right? Normally, this guy will hold on to oxygen, and you can see oxygen being held by the heme right there. Unfortunately, carbon monoxide is almost exactly the same shape as oxygen, and heme will also hold on to carbon monoxide. This is one of two ways that carbon monoxide kills you. One is by gobbling up the heme oxygen binding sites, and therefore your blood can't carry much oxygen. And the other we'll talk about later, it actually relates to energy. Okay? So carbon monoxide kills you in two ways. Okay, notice, oh, by the way, notice I said carbon monoxide, not carbon dioxide. Carbon dioxide is also poisonous, but for different reasons, and not because it is bound by the iron. It's not, okay? It's not bound by the iron at all. All right. Well, before I go on and talk about the next structure and then spend the rest of the lecture talking about hemoglobin, I want to say words about two processes, all right? One process is called denaturation. Denaturation. So what does denaturation mean? Well, from the perspective of a protein, denaturation means that you are unfolding the protein. Remember I said that the folding was critical for its function. If we unfold that, it will no longer have the same shape, and as a consequence, it will no longer have its function. Okay? This shows an interesting enzyme. We'll talk about enzymes later. Enzymes catalyze reactions. And this enzyme is called ribonuclease. And this ribonuclease turns out to be pretty stable to most of the things that we would use to unfold it. Well, how would I unfold? How would I denature a protein? One of the ways I can do it is by heating it. Heating it provides energy, and energy will break hydrogen bonds. So we could imagine if we heat a protein, we could change or unfold or unwind an alpha helix, for example. Another way that we can denature a protein is by adding detergent. So when we cook our food, the reason we cook our food is we kill bacteria, and we kill bacteria by denaturing their proteins. We wash our hands so we can eat our dinner because the detergent that's in soap denatures proteins and also kills bacteria. So it's this denaturation or the denaturing of proteins that's very critical for us to not get infected with things. Okay? There's other ways of denaturing proteins. Strong acids and strong bases will do that. Why? Because they drastically change the charge, and we've already seen how charges repel or don't repel or attract, and we might change that environment that's giving rise to stability of a protein. If we make it unstable, it's going to unfold. Now, I said this protein, this enzyme, was unusual in that it's actually fairly hard to unfold this protein. This protein has, at very strategic places within it, disulfide bonds. Anybody remember what disulfide bond is? How do we make a disulfide bond? What's that? It's a sulfur bridge. Two sulfurs. Two cysteines come together. We'll make a disulfide bond. That is a covalent bond. A covalent bond is strong. And these turn out to be very strategically placed in this protein so that even if I boil this protein as it is, when I reduce the temperature, these things are still in place, and the alpha helices and so forth can come back together, and this protein will come back alive, as it were. It'll come back to being active. Most proteins won't do that. And even though they have disulfide bonds as well, they're not as strategically oriented as these are. So if I want to denature this protein, I have to do two things to it. Boiling it alone will not unfold the protein. I first have to break the disulfide bonds, and then I boil it. Okay? And that's what's going on here. Oh, by the way, urea has the same effect as boiling. It's a chemical way of unfolding a protein. Okay? Urea has the same effect as boiling a protein. The beta-mercaptoethanol is a chemical that will break disulfide bonds. So you can see that the treatment of this with beta-mercaptoethanol has caused the blues to come apart, caused the purples to come apart, the greens to come apart, etc. And consequently, we have this structure over here. When urea comes along, it now completely unfolds everything, and this protein is inactive. Most proteins, if I simply boil them, hit them with detergent or whatever, they will come apart and will not be active. That's how we're killing bacteria. Okay. Now, folding of a protein is important, and it turns out that ribonuclease can be refolded. Okay? It can be refolded. If we completely take apart ribonuclease and we unfold it, ribonuclease has another odd property. And that other odd property is related to what I've already told you, which is that it will, in some cases, come back and completely make a functional enzyme. Most enzymes will not do that. Once we've unfolded them, they will not, under any circumstances, come together. But this guy, after we've unfolded it, will come back together and make a normal ribonuclease, a functional ribonuclease. What that tells us is one very important thing. And it's something I've already told you was the truth, but now you're seeing it demonstrated. And the truth is that the primary structure determines everything. It determines the properties of the protein, and one of the properties of the protein is the folding. Now, you might wonder why all proteins don't have this property, and it's actually a complicated answer to that question. The brief answer is because most proteins start folding as they're being made, and if you unfold them after they've been made, then you get interactions that don't normally happen in the folding process while they're being made. Okay. So don't worry about that. My only point of showing you this slide is just to convince you that the primary structure of a protein dictates everything else, including the folding. Okay. Well, I don't want to make too much of a point of this. This is not a big point, but it is an important point. Primary structure determines the other properties of a protein. What I want to spend some time now doing is talking about a really fascinating protein called hemoglobin. Hemoglobin introduces the last level of protein structure. We've seen primary, we've seen secondary, we've seen tertiary, and now with hemoglobin we're going to see another structure called quaternary. Quaternary structure relates to the interaction of subunits of a protein with each other. I told you last time that hemoglobin had 4 subunits. Only proteins that have more than one subunit will have tertiary structure. Only proteins that have more than one subunit will have tertiary structure. Myoglobin only has one subunit. Myoglobin does not have tertiary structure. Question? Sorry. Quaternary. Thank you. Okay. And now all over YouTube land, everybody goes, he said tertiary. Okay, it's quaternary. This is where I should just edit that out, right? So only proteins that have multiple subunits will have quaternary structure. Myoglobin does not have quaternary structure. Hemoglobin does have quaternary structure because it has 4 subunits. Now, it turns out that these 4 subunits give hemoglobin some amazing properties. And there's some amazing properties that myoglobin does not have. Okay? This is what hemoglobin looks like, and I want to say a couple things about this. Hemoglobin has 2 units that are identical called alpha. Okay? And it has 2 other units that are identical that are called beta. So it's got alpha 2, beta 2 structure. That's what we call it. And each alpha subunit looks very much like each beta subunit. They're not very different from each other. In fact, for our purposes, they're essentially identical. Moreover, the alphas and the betas are very similar to myoglobin. So all of these guys are related to each other. That's why we call them globins. Myoglobin, hemoglobin. And the globin refers to the protein part of these things. Alpha 2, beta 2. Because this looks like myoglobin, each subunit of hemoglobin contains a heme. There's a heme in green, there's a heme in green, there's a heme in green, and there's a heme in green. Hemoglobin has 4 heme groups because it has 4 subunits, each subunit containing 1 heme, and each heme contains 1 iron, and each iron can hold on to 1 O2 molecule. So hemoglobin can hold on to 4 O2 molecules. All right. As I always tell students, you know, that we can't do biochemistry without having a graph. We have to have a graph, all right? Now, this graph is fairly simple, but I will tell you that when you go to do graphs, this is true whether you're doing a titration plot or you're drawing a graph like this, any graph that you draw has to have the axes labeled properly or it has no meaning. What students tend to do is memorize shapes and forget what's giving rise to those shapes. Axes are critical. In the case of a titration plot, we saw it was pH versus OH added. If you just drew a titration plot, it wouldn't have any meaning because the axes aren't defined. That could be referring to atomic energy for all anybody knows, right? Okay. In this case, we're plotting on the y-axis what percent of the molecules are full of oxygen. That's what's on the y-axis. On the x-axis, we're plotting basically the pressure or the concentration of oxygen. That's the 2 things that's being plotted here. Now, if we look at these 2 plots, one for myoglobin and one for hemoglobin, we see that they're very different from each other. Okay? Myoglobin goes up very steeply and then bends over like this. Hemoglobin, on the other hand, has a curve that we call sigmoidal. It looks like an S shape. Okay? We call this hyperbolic. That's the shape of the myoglobin. We call the hemoglobin's curve sigmoidal, meaning it's like an S. Well, what does it mean to have these different shapes that are there? Well, let's think about this. Let's look at the myoglobin first. If we look at the myoglobin, we see that at very low oxygen concentrations, this thing is 50% saturated. Meaning what? It means that myoglobin is very good at grabbing a hold of oxygen and hanging on to it. Only when the oxygen concentration gets really, really low does myoglobin let go of its oxygen. Myoglobin functions in our muscles, that's the myo part of that. And in our muscles, myoglobin is what I like to describe as an oxygen battery. An oxygen battery. Your laptop has a battery in it. That battery can let you go into biochemistry and sit down and type notes, as yours is doing right here. And when you go to plug it in, it remains on. You unplug it, it stays on. The battery fills in as it needs the power. An oxygen battery does the same thing, but in the case of myoglobin, it really only gives up its oxygen when the concentration gets very low. And we'll see later that's important for muscles. Because it's possible for you to exercise more rapidly than your blood supply can bring oxygen. You can use oxygen in your muscles faster than your blood can supply it. Having a battery that fills in that gap when the oxygen concentration gets low turns out to be very useful. And that's why myoglobin is found in our muscle cells. Myoglobin is great at holding on to oxygen, but it's not so good at giving it up during normal conditions. Okay? So we would be very happy if we had myoglobin grabbing oxygen in our lungs, but we wouldn't be so happy when it got out to the tissues, because only when the tissues were starving would myoglobin give up its oxygen. So myoglobin is not so good at carrying oxygen. Hemoglobin is fantastic for it. Here's why. Okay? Hemoglobin, if we get out here and we were to take this plot and go all the way over here, we would see that these two lines would intersect about right up here, way over here on the right. They would intersect. And at that concentration of oxygen, that would correspond to the concentration of oxygen we would have in our lungs. At the concentration of oxygen in our lungs, we have essentially 100% of the hemoglobin bound with oxygen. We'll see how that happens in a minute. But essentially 100% of the hemoglobin will have oxygen, just like 100% of the myoglobin will have oxygen. As you're sitting here right now, the concentration of oxygen in your tissues is about right here. You can see that at this concentration of oxygen, hemoglobin is giving up about 60% of its oxygen. Your cells aren't having to starve for oxygen to get it. Hemoglobin is providing that oxygen for them at a reasonable concentration of oxygen. They don't have to go down too low to grab it. That makes sense? Questions about that? No questions? Should I give you a pop quiz about it? Are there questions now then? Yeah. Well, okay. So if we look at the concentration that normally exists in our tissues, as we're just sitting here, like you guys are sitting here, that's roughly in this area. Well, if we look at what percentage of the hemoglobin that is bound to oxygen, it's roughly 40%. meaning that as the hemoglobin is traveling through the body where the oxygen concentration is the normal concentration for tissues, 60% of the hemoglobin has given up its oxygen and 40% is still holding on to it. What we want hemoglobin to do as it's traveling through our body is pick up oxygen in the lungs where the concentration is high and drop off oxygen in the tissues where the concentration is lower. This works really well. Now I'm going to explain to you how this works. Okay, how does it work? Well, the first thing I want to tell you about is something called BPG. Okay? I want to come back to the structure of hemoglobin. I meant to mention this earlier. If you look at the structure of hemoglobin, you'll see that hemoglobin looks like a donut. It looks like a donut, right? It's got a hole in the middle. And the hole in the middle of this donut is very critical for the function of hemoglobin. I'd like you to imagine that I am a fast jogger. I'm a slow jogger, actually. But let's imagine I'm a fast jogger. And I go out and I start running, as I like to run in the mornings. And I go out running, and I start flexing my muscles. Muscular contraction requires energy. And energy requires oxygen, as we will see later. Okay? Those muscles would like to send out a signal saying, hey, buddy, can you dump a little bit more oxygen here because here is where we need it. Remember, we had about 40% to play with there, right? Here's where we need it. Well, it turns out that the body has several ways of signaling where the oxygen is needed. One of these is a molecule called 2,3-BPG. How many people in here have heard of 2,3-BPG? It's rare. You've heard of it. Okay, good. Have you watched my videos before, or did you hear it elsewhere? Exercise, sports science. Okay. 2,3-BPG is a fascinating molecule. We'll talk about it later in the term, but it's actually made made as a byproduct of rapid metabolism. It's a byproduct of sugar metabolism specifically. And 2,3-BPG has this really cool property. It fits right in the middle of that donut hole really well. So what? Who cares? Well, here's who cares. It turns out that hemoglobin has two different structures. That is, two different ways for these chains to interact with each other. And they're very, very subtly different. Slight change in shape, as we're going to see, that's caused by the binding of 2,3-BPG has a very big effect on hemoglobin's properties. Everybody with me? Okay. The effect that the binding of 2,3... Let me show you what 2,3-BPG looks like. Okay. So 2,3-BPG, same structure, looks like this. No, you don't need to know the structure, but it's a small molecule. Fits in that region pretty well. Hemoglobin, I told you, had two structures. One structure is called the R state, and the other one is called the T state. R state and T state. The R state is a state of hemoglobin that likes to bind to oxygen. When hemoglobin is in your lungs, what happens is, let's trace a hemoglobin going through your body. Hemoglobin has gone through your body, it's dumped all of its oxygen, let's say. It gets back to the lungs, and it encounters a very rich oxygen environment. It turns out that when hemoglobin gets back to the lungs, it's in a state we call the T state. We'll see why that happens in a second. T state stands for tight. And just like a person who's uptight, it's not very flexible. And because it's not very flexible, it does not bind to oxygen very well. So when hemoglobin arrives in the lungs, it is not in a structure that allows it to bind to oxygen very well. Everybody with me? Remember, there's 4 subunits. The binding of oxygen by one subunit causes hemoglobin to change from the T state into the R state. What does that mean? Well, in the R state, that's the state that's flexible, and it likes to grab oxygen. So the binding of one, even though hemoglobin didn't want to bind that first one, when it binds the first one, the molecule changes shape, and now it grabs all the other oxygen. That turns out to be very important. This is how hemoglobin gets loaded up with oxygen as it's traveling through your lungs. You want that to happen fast because there's not a lot of time for hemoglobin to grab oxygen. Binding of one favors the binding of everything else. That phenomenon is known as cooperativity. Cooperativity occurs when the binding of one molecule favors the binding of other molecules. So we see how hemoglobin has changed from the T state into the R state. The R state likes to hold on to oxygen because it's been grabbing it out of the lungs. It likes to hold on to oxygen. Let's follow this hemoglobin now through the body. It's left the lungs. It goes down to my legs, which are exercising. And my legs, which are exercising, put out 2,3-BPG because that's a signal that they are going through rapid metabolism. Hemoglobin binds to oxygen. Hemoglobin binds to 2,3-BPG. And when it does that, it converts from the R state into the T state. That has the effect of causing hemoglobin to let go of oxygen. The signal is 2,3-BPG. That's really cool. The muscles are saying, hey, we need some oxygen right here, right here, right here. Here's our 2,3-BPG. Hemoglobin binds to the 2,3-BPG, and oxygen gets dumped right there where it's needed. But there's a problem. Hemoglobin has just bound something that caused it to turn into the T state. If it doesn't get rid of that 2,3-BPG before it gets back to the lungs, it's not going to be able to flip into the R state when it's in the lungs because the 2,3-BPG will hold it in the T state. Well, it turns out that 2,3-BPG does not bind covalently. Does not bind covalently. When things don't bind covalently, they can come off, come back on, come off, come back on, etc. And so as hemoglobin is making its way back to the lungs, 2,3-BPG comes off, comes on, comes off, comes on, comes off, and sometimes when it comes off, a cell grabs it and uses it for energy. 2,3-BPG is one of many foods for cells. So now you see how hemoglobin dumps its 2,3-BPG. It's still in the T state, but it's not locked in the T state. 2,3-BPG locks it in the T state, but now it's just sitting here in the T state waiting to bind to an oxygen so it can flip into the R and bind more oxygens. It's a beautiful system, an absolutely beautiful system. Grabs oxygen in the lungs, 2,3-BPG binds, causes hemoglobin to let go of oxygen, 2,3-BPG disappears before it gets back to the lungs. Everything is cool. Except there's a problem. The problem is if you smoke. I hope none of you smoke, but if you're a smoker, here's a big problem. If you are a smoker, your blood has more 2,3-BPG in it than anybody else's blood does. If your blood concentrations of 2,3-BPG are high, what does that mean? It means that when that comes off, it's going to be more likely that one's going to come back in. It means it's more likely that hemoglobin is going to make it back to the lungs, bound to 2,3-BPG. It's going to remain locked in the T state. Your body is going to have a reduced oxygen carrying capacity because Hemoglobin is going to be locked in the T state in the lungs. It's going to get very little oxygen, and this is why smokers get out of breath when they climb stairs. One molecule, some very drastic effects. And these effects are happening as a result of very tiny changes in the structure of hemoglobin. The T state and the R state, if you were to look at them with your eyes, you would have a hard time distinguishing them. They're very subtle changes, but they have very drastic effects. Questions about that? Yes? Yep. That's the most common question I get. Why do cells have more 2,3-BPG? I'm going to ask you to hold that question until we talk about sugar metabolism. There's a very good reason why, but part of it is because smokers have carbon monoxide in their blood. And the carbon monoxide is actually a big contributor to that problem. But let's save it and talk about that then. That's okay. Okay? And that's the most common question I get. Was that your question also? Yep. Yep. All right. Okay. Yeah. Of 2,3-BP of smoking?
Yeah. So let's say
yeah.
a child who is like a hand smoker, are they going to have
yeah.
the effects of it later in life as an adult?
So the effects of 2,3-BPG go away fairly quickly for a smoker. So when we think about effects of smoking, they happen at many levels. The cancer effects and so forth can persist throughout lifetime. All right? But the 2,3-BPG effects can go away fairly quickly. Yep. It's one of the first benefits people notice when they stop smoking is they have more aerobic capacity. Yep. Yeah? So the question is, as you exercise more and you build up more endurance, what's going on, basically, is what you're asking. Okay? So there's several things physiologically that can happen. One of the things that can happen is you can start making more hemoglobin, which is going to increase your oxygen carrying capacity. Your body has an ability to adapt to a variety of conditions like this. If you ever go and live at a high altitude, I had a student one year who went and to the Andes. He was living in Ecuador. And so he was living at close to 10,000 feet, okay? And he was a jogger. He and I used to jog all the time, okay? And when he came back after being gone a year, the first few days that we jogged together, it was like there was no way I could keep up with him because his body had adapted to the lower oxygen environment and had made much more hemoglobin. And so he was in great shape. It didn't last forever, about a month or so, but it finally wound back down. But your body does adapt to those. So that's part of what's happening. Okay. Other questions? All right. Well, hemoglobin, as I said, is a remarkable molecule. That's one of many things that it does. I'm going to tell you some more. Okay? I'm going to tell you some more. Another effect is called the Bohr effect. And that's not what I wanted. The Bohr effect, okay, is shown right here. Now, ignore the myoglobin for the moment. All right? Myoglobin just confuses the picture here. What we see are the binding curves of the same hemoglobin at different pHs. So the pH of your blood is about 7.4. We see that if we change the pH of the environment in which the hemoglobin is found, its oxygen binding properties change. Basically, curves that go more to the left have stronger affinity for oxygen, meaning they will bind more oxygen, and those that move to the right will have less oxygen binding. As the pH gets lower, you see hemoglobin tends to let go of more oxygen. This also is a signal. Rapidly metabolizing tissues, like my muscles, release protons. It changes the pH in the region of where that muscle cell is. The protons bind to hemoglobin. What do you suppose the protons would do to hemoglobin? Hopefully you would tell me that they would bind. They're charged. They would change the charge environment. They would slightly change the structure of hemoglobin. And that slight change of structure of hemoglobin would cause oxygen to be released. And that's exactly what happens. Okay? Again, there's another signal. The signal in this case being protons causing hemoglobin to let go of oxygen. Okay? There's even more. All right? Even more. Oh, actually, it's not on here. All right. If I were to do this same set of curves, and let's say I took 7.4, this guy right here, this sort of bluish one, which is about the pH of your blood, and I did another binding curve, and I did it at 7.4 also, but I added some carbon dioxide, I would see curve shift to the right again. Meaning that carbon dioxide will favor hemoglobin letting go of oxygen. And this turns out to be a third signal. Rapidly metabolizing cells release carbon dioxide. And notice I said dioxide, not monoxide. Rapidly metabolizing cells release carbon dioxide, and carbon dioxide can also bind to hemoglobin, not at the heme group, it binds it elsewhere, but the binding of carbon dioxide onto other portions of the hemoglobin molecule cause it to change shape slightly. That slight change in shape favors the release of oxygen, and now we've got our third thing that causes hemoglobin to let go of oxygen, and that third thing is all three of them are found at rapidly metabolizing tissues. Remarkable. Okay? Hemoglobin's not that big, but it responds to all of those things. Okay? But wait, there's more. You're getting tired of that, aren't you? There's actually more. Okay, what was this? This was BPG. Okay, all right. Fetal hemoglobin. All right? It turns out that fetuses have a slightly different hemoglobin than adults do. It's called fetal hemoglobin, and it turns out to be really important because the fetus has to take oxygen away from mom's hemoglobin. It has to take oxygen away from mom's, which means that ideally the fetal hemoglobin will have a greater affinity for oxygen than mom will, than mom's hemoglobin will. Okay? Here's mom's hemoglobin. Fetal hemoglobin, the same curve. We see it shifts to the left, which means greater binding, which means that the fetus is able to take oxygen away from mom's hemoglobin. How does it do that? All right? Well, I'll tell you. First, the fetal hemoglobin I said was slightly different. Instead of having alpha 2, beta 2, it has a related set of units called gammas. Instead of the betas, it has 2 gammas. So fetal hemoglobin is alpha 2, gamma 2. They're very, very similar, but they're not identical. It turns out that the addition of the 2 gammas changes the shape of the donut hole. It changes the shape of the donut hole. Because of the change of the shape of the donut hole, the fetal hemoglobin cannot bind 2,3-BPG. The fetal hemoglobin cannot bind 2,3-BPG, and the net result of that is that the fetal hemoglobin stays mostly in the R state. The R state was the greater affinity. So the fetal hemoglobin is in the R state more than mom's is, and that's how the fetal hemoglobin takes oxygen away from mom. Pretty cool. There's even more. Everybody's heard of sickle cell anemia, right? Sickle cell anemia results from a mutation in hemoglobin that causes the hemoglobin to form polymers. When it forms polymers, the cells contain it stop being round like a nice blood cell, and instead becomes sickle-shaped. That's where the disease gets its name. So you can imagine you've seen the nice pictures of red blood cells that are nice, rounded, sort of ovoid structures. I don't have one on here to show you, unfortunately. But it has a smooth shape. And that turns out to be really important because blood has to travel through capillaries. That's where most of the oxygen exchange actually occurs, is in capillaries. That smooth shape allows a blood cell to navigate through those capillaries and not get stuck. If you have sickle cell anemia, what happens is under low oxygen concentrations, the polymerization happens and the sickling of the blood cell happens. And when a sickled blood cell tries to make it through a capillary, it gets stuck. It gets stuck. It's a very painful condition if you have it. It means when you exercise and you have sickle cell anemia, your muscles are not getting the oxygen that they need. It can be very painful. In some cases, it can be life-threatening. Okay? And it's happening because of mutations inside of hemoglobin. Okay? I told you there was a lot to hemoglobin. A lot to know there. Let's finish it with a song. What do you guys say? This is a very popular song. It's called Hemoglobin is Moving Around. I want to hear you today, too. Oh, isn't it great what proteins can do, especially ones that bind to O2. Hemoglobin's moving around. Inside of the lungs, it picks up the bait, changes itself from T to R state. Hemoglobin's moving around. The protoporphyrin system, its iron makes such a scene, arising when an O2 binds, pulling up on histidine. The binding occurs cooperatively thanks to changes quaternary. Hemoglobin's moving around. It exits the lungs, engorged with O2, in search of a working body tissue. Hemoglobin's moving around. The proton concentration is high and has a role. Between the alpha betas, it finds imidazole to empty their loads. The globins decree. We need to bind to 3-DPG. Hemoglobin's moving around. The stage is the set for grabbing a few cellular dumps of CO2. Hemoglobin's moving around. And then inside the lungs it discovers oxygen and dumps the CO2 off to start all over again. So see how this works. You better expect to have to describe the Bohr effect. Hemoglobin's moving around. All right.